Start 3 seconds early.
Alright. Hi, I'm Craig.
Hi, I'm Margo.
And I'm Michelle, and we're here representing the Boston University Wellesley College software team. So actually the three of us who will be presenting today represent the three different portions of our team. Margo is here representing the wet lab part of the team. Craig is representing the back-end algorithmic part of the team. And I'm here with the front-end human-computer interaction part of our team. So when we started this summer, we thought about what sort of challenges the synthetic biology researchers face. And we realized that they fall into three main areas: manipulation and maintenance of large amounts of data, designing biological constructs based on that data, and then execution of those designs in the wet lab. And there are lots of sub-problems that arise within each of these areas. And on analyzing them, we realized that there are three main places where we think we can use computation to help out synthetic biology research teams. And those three areas are characterization of biological parts, collaboration between wet lab users, and dealing with the complexity of the problems that synthetic biology researchers face on a daily basis. So we looked at this and we said, we could help out in any one of these areas, the data, the design, the execution, but just doing one of them isn't going to be enough. If we want to drive synthetic biology research forward, we need to do something that will take care of all of these areas, which is why we created a suite of software tools that we think addresses all parts of the synthetic biology workflow. Our vision for this summer was that we wanted to apply engineering principles to synthetic biology by utilizing the user-centered design to build software tools that foster collaborative problem solving, support complex system design, and enable automation in the lab. So during this talk, we're going to talk about 5 different software tools that we created over the summer. And then afterwards, we're going to talk about how those tools were actually applied in the wet lab portion of our team and what feedback they had about those tools. The thing that unifies all of these tools that we created is Clotho, which is a data management tool that's unified by a core data model. And this tool was created by Doug Densmore's iGEM teams in 2008 and 2009. If you want to find out more information about it, you can check out our wiki. We have links to all of that information. So for all of the software tools that we created this summer, we utilized what's called a user-centered design process. And the idea behind user-centered design is that we want to get users involved in every aspect of software creation, starting with brainstorming, moving through to creating our first designs and prototypes, getting users involved in giving feedback on those prototypes, through increasing levels of fidelity, moving from paper prototypes to actually coded implementations of the system. So that sounds like something that's pretty obvious, right? You want users to be involved in the product that they're eventually going to use. But it turns out to be a lot more work than you would anticipate. We started out this summer with observations. So the computational part of our team spent hours in the wet lab watching our wet lab members go about their daily work of doing synthetic biology research. And from that, and we also visited the MIT iGEM team to find out what sorts of problems they faced that might be different than our own team. From that, we figured out what sorts of problems we thought we could address using our computational abilities. So the first tool that we want to present to you today is called Genome Surfer Pro, which is a tabletop interface for collaborative prokaryotic genomic data exploration. So the driving force behind this application is that while an iGEM team might start out the summer with an idea of what organism they want to research, they don't necessarily know which genes in that organism are relevant to their specific research question. Or even if they do, there's more background research that they need to do before they can finalize that decision on which genes they want to use. So the Wellesley Human-Computer Interaction Lab had already addressed some of these challenges with eukaryotic organisms with a tool called Genome Surfer. And this summer we leveraged our experience creating that system to build from the ground up Genome Surfer Pro, which is the same sort of system but for the exploration of prokaryotic data. So as a synthetic biology researcher, you come up to this table and select what sort of organism you're interested in looking at that day. And it shows you a circular representation of the circular genome of that organism, and also a bar at the top of the screen with sort of the region around the gene that you're looking at the moment. When you find a gene that you're interested in, you can pull up DNA and protein sequences for that gene. You can search PubMed for publications on that gene or on the entire genome. And you can perform a blast against the entire NCBI database, all within this application. So there are several benefits to this. The big one is that this system will promote collaborative hypothesis forming. We chose to implement this system on the Microsoft Surface, this tabletop touchscreen that you see. And the reason behind that is that work on the Surface is inherently collaborative. When you have a traditional keyboard and mouse, then there's only one person who's sitting at the computer, one person who's manipulating the data directly. And our thought is, when you have an iGEM team, when you have multiple people working on a problem, it's better to let everyone sit around the table together and to literally get their hands on the data. Another tool that we created this summer is called Optimus Primer, which is a multi-platform collaborative primer designer. So I know that we've seen lots of primer designers at iGEM, so you're probably thinking, why do we care that we've made another one? So there are two main reasons that our primer designer is really exciting. One of them is integration with Clotho. So our primer designer, anytime that you design a primer, you can save it to Clotho as an oligo, which means that you can easily share it with other wet lab members or with other iGEM teams or other synthetic biology researchers in the community. The other really awesome thing about our primer designer is that we implemented a version for the Microsoft Surface, which is integrated with Genome Surfer Pro. And that system is useful because it's on the Surface. It has the same sort of collaborative properties, which means that when you're first learning to design primers, you can sit down with a mentor and work together to figure out how primer design works. And when you're first learning how primer design works, it's very complicated, so it's nice that you can both manipulate the primer design process at the same time. So next we have a short video to illustrate these two applications. We'll be using videos throughout our talk to show some of the applications we created this summer because we couldn't bring the Surface here to Indianapolis to show you in person. If you come by our poster tonight, we'll have laptops with demonstrations of the PC applications that we made this summer, and we will have the Surface at the World Jamboree if you're around then.
So now we know what genes we are interested in studying, and we can effectively bio-break these genes, but we're actually interested in how these parts interact with one another. So let's imagine that we're interested in 5 different parts. If we want to study a system composed of these 5 different parts, we first need to characterize each subset of these parts. This means building many different plasmids with many different combinations of these parts, which does not sound fun for our wet lab. So what if instead we could put all of these parts into a single plasmid, and then through some actuation mechanism, be able to selectively turn on and off some of these parts? Then we would only need to be able to build a single plasmid, and through this actuation mechanism, we could make any of the combinations that we want to characterize. So you might say, this sounds good, but how exactly do you propose that we do this? So the answer to this actuation mechanism comes in the form of an interesting enzyme called an invertase. So how does an invertase work? Well, these special enzymes recognize recognition sites within a duplex DNA, which are oriented in a proper position, and then they effectively invert the DNA region between the 2 sites. So for example, here we see a part which is surrounded by 2 invertase sites located, or pictured here by these green triangles. And then when we input the corresponding invertase, we see that this part is effectively flipped. Now you can imagine that if we use this with promoters, we can select which genes to turn on and off. Not only that, but we can also permute these different parts, which is a new and exciting problem to solve. So here we have an example of a 2-part fully reconfigurable circuit which uses 3 distinct invertases. The first thing to note is that we have 2 different parts, and each one is flanked by its own set of invertase sites, here depicted in the orange and the blue triangles. So if an invertase is inputted to the system which corresponds to these sites, then these genes can be turned on and off selectively. It's also important to note that the entire set of genes is flanked by these red sites here on either end. So if we input this invertase to the system, then we are able to permute the 2 genes. So this is a pretty simple construct, but you can imagine that as we scale the number of parts up, the placement of invertase sites so that we can hit all of these permutations and combinations is not a trivial task. Due to this, there needs to be an algorithm developed which properly places these invertase sites. So for example, here we see a 4-part system, and you can see that the complexity of these invertase sites grows very quickly. So this summer, we not only designed 2 different algorithms which properly place these invertase sites so that you can hit your desired permutations and combinations, but we automated the entire design process with a Clotho application called Trumpet, which is a play on the word permute. So how does Trumpet work? Trumpet, how does Trumpet work? Trumpet allows the user to select what parts they're interested in permuting, and then the user is allowed to choose the permutations and combinations they're interested in achieving. Then the software uses the algorithms that we developed this summer to properly place the invertase sites so that these permutations and combinations can indeed be achieved. Along with the design, a list of keys is returned to the user, where a key corresponds to the sequence of invertases that need to be inputted to the system so that that combination or permutation can be achieved. So again, we have a short video for you which shows this off. I do want to mention actually that we have 2 versions of this. One is a Clotho application and one is a Surface version that we're currently working on. So far we have shown you software that allows us to choose what parts we're interested in characterizing, and we can biobreak these parts, and we can even make reconfigurable genetic circuits out of these parts. But what about building these parts? Well, this summer our team spent a lot of time trying to be able to automate this building process. And as a result of this time spent, we came up with a Clotho application called Puppet Show. Puppet Show allows us to automate lab protocols. So how does Puppet Show work? At a high level, Puppet Show starts with a specification, a design specification. This would be the output of something like Trumpet or Optimus Primer. And in the end, Puppet Show communicates to a liquid handling robot and actually builds this construct. So this overall flow can actually be split up into a number of different stages. At the top, like I said, we have the high-level specification of our part, or our construct. And then this construct is then divided into its primitive biological parts. So these are your biobricks, for example. And using the Clotho API, we come up with an optimal plan for assembling all of these different parts. Then this plan is translated into a set of protocols. These are your ligations or your restriction digests. And these correspond to a set of Puppeteer code for each of the protocols, which is then compiled by our Puppeteer compiler and eventually sent to the vendor-specific software for our liquid handling robot. On top of automating these lab protocols, we also wanted to make things run a little bit smoother in the wet lab. So we came up with this idea called the eLab Notebook. This is an iPad application, and we think that maybe in the future, if iPads are mounted in some way to your lab, then you could use this application to let the users of the wet lab know what's going on between one another. So it would show, for instance, what equipment is currently being used, who's running what protocols, you could leave notes for each other, and so on and so forth. I just want to note that this is still sort of in the design phase. We have this idea and we're sort of working on a prototype currently, but we don't have a working version to show, but I encourage you to go to our website and check out more of the details on how this is implemented. And again, we have a short video to show you this whole automation process.